Hello everyone. I'm Munib, a data scientist specialized in credit risk management and co-founder and the co-organizer at Tunis. So I'll be the moderator of this meetup today. Thank you all for joining us today to attend our meetup, which will be about integrating gene expression and DNA binding data using our bioconductor. So the workshop will be animated by Dr. Mahmoud Shaaban. It will last approximately 2 hours. And I want also to let you know about this workshop, that this workshop, in fact, is a part of the Bioinfo series that we have launched since September 2020. So Dr. Mahmoud Shaaban graduated from a medical school in Cairo University, Egypt in 2015. He received his master's and PhD from Gyeongsang National University, Korea in 2021, where he studied the role of autophagy in development and disease. He used our bioconductor to analyze gene expression and transcription data from mice models. He wrote also very nice open source R packages to distribute the data, methods, and code of his research. So he's so talented, so stay tuned. So now let me just briefly introduce to you our team, Tunis, through this video. So I will be sharing my screen. Okay.
Hello and welcome to this workshop session. Before starting the workshop, we'll introduce you to the Our Ladies Global and Our Ladies Tunis initiative. The R community suffers from an underrepresentation of minority genders in every role and area of participation, whether as leaders, package developers, conference speakers, conference participants, educators, or users. As a diversity initiative, the mission of Our Ladies is to achieve proportionate representation by encouraging, inspiring, and empowering people of genders currently underrepresented in the R community and to facilitate individual and collective progress worldwide. Gabriela de Queiroz founded Our Ladies on October 1, 2012. Our Ladies Global was born and the grant was awarded in September 2016. Since then Our Ladies has grown to 170 chapters in 44 countries and 39,000 members. Our Ladies Tunis is part of Our Ladies initiative and was created in May 2020 by women working as data scientists and biostatisticians and bioinformaticians. Our goal is to create an R community in Tunis and empower underrepresented genders in the R community. This is the core team of Our Ladies Tunis. Come and join our community on social media. This presentation was made with R Markdown. Thank you for watching this video and enjoy our workshop.
Thank you all for your attention. The screen is yours, Dr. Mahmoud.
Thank you so much for the introduction, and I would like to thank you for the effort that you're doing in Tunis to spread the word about R and data and data science. I'm really glad to be here today, and I want to thank you guys all for coming. It's the weekend in some countries and or in most, and I really appreciate that you take time of your weekend to come to listen to me talking. And I'm really looking forward to learn from you all and see if I can be of help. So I just posted the link in the chat. So this would be the website that we'll be working from. It's a package down website. So for people who don't have who can't run the code itself, you can just use this to see the code and to see the output of the code. And I will share my screen to start. Okay, so you guys can see the screen share?
Yes.
Okay, great. Okay, so today I want to talk about a workflow for integrating two types of data, of high throughput data. Those are gene expression data and DNA binding data. And most of the work uh, is done in R, and more specifically using bioconductor packages. So the idea here is that people who deal with ChIP-seq data can use it to identify transcription factor binding sites. And people who work with gene expression data, they can, if for example, you have a perturbation experiment where you knock down or overexpress one transcription factor, you can see the effect of this perturbation on the gene expression by saying the knockdown of this factor upregulated or downregulated the expression of that gene or of this gene or that gene. But I was looking for a way to integrate those two types of data so we can get high confidence, and if possible, we can get more information from the same set of data. So in this workshop, I'm using gene expression data from microarrays and DNA binding data from ChIP-seq from the same cell line to see if we can use the two types of data to predict the function of the transcription factors on their specific targets. And at the end, we'll try to see if there is an interaction between two transcription factors that work in the same cell line. Those two transcription factors are very related, so it would be interesting to see if they have shared binding sites, and if they do, what is the function of what would be their function if they bind to if they both bind to the same gene. So it would be very useful if people are familiar already with R and Bioconductor, but don't worry about that. We will go through the everything that we need to know to understand what's going on in this presentation. So there are different ways that you can follow through if you want to run the code, or you can just watch, or you can follow the pages that we're using on the website that I just posted in the chat. So one way to execute this code is to use RStudio Cloud, and it's free. Although because of the limitation in the resources, it might be a bit clunky and it might not work at all. So to do that, you can just create an account on RStudio Cloud, and you can create a new project. And when you create a new project, choose create a project or a new project from Git repository, and then post this link in it, and then it will pull down the code, and then you can install the packages and start running the code. If you want to run this locally, you can, in RStudio on your local machine, you can just clone the repository from here, and then just open the vignette in RStudio, and then you will be able to execute the code after installing the packages. The third option is to use Docker image. So this Docker image is ready, so you can just download it using the Docker pull, and then run RStudio server using this command. And then you will be able to use it just like you use RStudio on your local machine. If you had any troubles, so just to see if the trouble is from my end or on your machine, you can just run this cmd check, rcmd check command. It should execute without errors. Otherwise, there must be a problem with your personal setup. So at the end, also, we can spend a bit more time to see if people were actually able to run this code, and if there are any problems that I can help with, I'll be really happy to. So the Bioconductor packages that we're going to be talking about in this presentation is this mainly the target package, which is a package that I contributed to Bioconductor a year ago. And then there are some data management packages from Bioconductor and from R in general. And then there are a couple of annotation packages that we will talk about as well. So the outline, the time outline of this presentation will be like 20-minute sessions. I will first talk about bioconductor packages and classes that are relevant to this presentation. And then I will introduce the package that I developed. And then we will see a use case of two transcription factors called YY1 and YY2 in HeLa cells, and see if those two factors interact together. And if they do, what is the nature of this interaction? Do they cooperate or compete on their shared targets? So my goal in this workshop is to discuss with people how to use R and bioconductor to read in differential expression data and binding data, and then to run a predictive analysis, and then to look at the output and try to understand it. And then hopefully this would suggest, will either give you an idea about what the data is saying, or at least it would suggest what you should do next. So yeah, it's about reading in data, using bioconductor to tie together the different types of data, and then to prepare the data that you just read in the specific input format that the target analysis requires, and then run the target analysis, and then try to visualize the output and understand it. So I will be talking about using R to read in data frames from the gene expression data in data frames, and then use the bioconductor package, the bioconductor structure R ranges to read in the bid files from the ChIP-seq data, and then how to extract the annotation from the taxonomy and the organism packages, the annotation packages, and then how to apply the target analysis that I presented in this package, and using some of the helper functions in the package, how we can test and visualize the output. Okay, so in this part, I want to talk about which bioconductor packages are relevant to this kind of analysis. So a brief introduction to the bioconductor itself. For people who are not familiar with it, it's basically a collection of R packages that have certain guidelines and certain data structures that they use and reuse, and then people can specifically designed for genomics data analysis and transcriptomics, but also other biological data. Around this project, there is the Bioconductor organization itself that does much more than just the developing packages, such as outreach. They organize conferences, and they do a lot of interesting stuff. So I would advise you, if you are interested in bioinformatics or computational biology, to check out the Bioconductor pages. So the Bioconductor has three different types of packages. The first one is software packages, and these are either infrastructure and data structure packages, or they are analysis tools. The second type is annotation packages, and these are specific kinds of data about genes, about mRNAs, about proteins, that are not they're not data that you generate in the lab every day. So like gene identifiers or gene samples would be an example of these annotations, as we will see later. The last type is experiment data or experimental data packages, and these are usually data sets that people use to test their packages or to visualize their methods that they develop. So on the software packages, more specifically, either implements algorithms or give you access to resources, bioinformatics resources like NCBI or biomarts. And visualization is really important when it comes to genomics and transcriptomics data, because usually you have a lot of data that you want to show in a single plot. So you really need to think about visualization, and Bioconductor packages are really helpful for that. The annotation packages are, again, it's usually in the form of a database, and then the package will give you an interface to this database. Gene samples would be one example of this, of this kind of information. the gene coordinates, like the chromosome names or the start sites of specific regions, is also you can also get from these kind of packages. The final part is the experimental data packages, and usually these are just like small data sets that people make available to test their workflows. But there are also there are a lot of people also who contribute their own data in the form of experimental package, so that they can increase the utility of it by making it available and other people can use it. So the idea of building a standard or a class to visualize to contain or data containers for genomics data comes from the need that usually have different types of data and a lot of it at the same time. So if you just write it to a text file and try to work with it, you need to know exactly how the data is represented in the file. So if it's your own file, that's fine because you wrote it and you know how it works. But if you want to share this with other people, it would be really difficult. So the Bioconductor team came up with something called the three-table structure, where you can have different types of data and they are tied together in a very formal way. And then this is also a standard, so everybody is using it. So if I give you this data structure, you would know exactly what to do with it, or like where is every type of data located in the structure. So the simplest three-table structure would be, I can explain it by using an example. So for example, if you have RNA-seq data, so that your main data would be the gene expression, the gene expression or the read counts. So this would be this table in the middle. So we put the samples by convention. We put the samples in the columns, and then we put in the rows we put the features, and then you add another table for the phenotypes data or the samples data, and this would have the same. That would have a key column, which is the column names of the expression matrix, and then you do the same for the features. For example, those could be genes. And then if you subset this data, this part, it will automatically propagate into the object. So it makes it really easy to add a lot of data and to add different types of data at the same time. So one example of the three-table structure would be the summarized experiment object, which we'll talk about later. So the Bioconductor provide two main types of functions to interact with data structures. So the first one would be a constructor function, where it's usually the name of the object, and then you put the pieces of the object into as arguments. And then it also provides accessors, which is usually the name of the specific piece that you're looking at. So for example, here, the gene expression, the gene counts matrix usually is put here in something called SA. And then you add it to the object, and then you access it using the same function name. So another key package in the Bioconductor is the genomic ranges, which is really important for people who want to use Bioconductor to analyze genomics data or transcriptomics data. So the idea here is that we want a formal way and a standard way to represent the locations of regions, such as genes or promoters on the genome. So we do that by identifying or by specifying the start site and the end site. And then we can add names for that in something called I ranges. So if you do this simple example, what you get is an object called regions. The class of this object is called I ranges. And then it has a column that says what the start is, what the end is, and then automatically it calculates the widths of these regions. Then you can expand this a little more to have a G ranges. The G ranges is basically an I ranges object, so just like the one we just created. And then you add a sequence name, which is the chromosome name. Usually this is the chromosome name. And then this object would look like the I ranges, with the difference being that you have a sequence name now. And then you can add more and more information using using these standard inputs, such as you can add a strand for these regions. So this is also this is again another example of how you create the GRanges object, and you can immediately see that this can be extended to this can be very powerful because you can represent data in a formal way and you can share it between people, and then you can name it. So an example of the accessors that I just talked about is the seq name. So a seq name on the GRanges object gives you the sequence name. Start will give you the start site, end will give you the end site, and then strand will give you the name of the whether it's a positive or a negative strand. But there is more than that. So you can compute on these objects because the data is represented in a standard way. You can compute in these objects. So for example, you can write a simple function called widths, and what it does is subtract the end from the start, the end side from the start side, and give you the widths. You can also say that this simple idea can be extended to functions that does a lot of calculations. And then for the GRanges object, the last thing is you can add metadata. We call it mcalls, and these are just data frames of arbitrary lengths. They just have to have the same row number. So for each of these regions, you can add a region name. You can add any other information that you want to add. So the example of the three-table structure is the summarized experiment object. This is pretty much the most commonly used. And I'm trying here to explain how you can use this three-table structure to like code your experiment or like to formalize the experimental design that you're using. So for example, here, if we have 4 columns, each is just called sample 1 and sample 2. We have 2 controls, we have 2 treatments. So you can just make a simple data frame with that. And this would be the phenotypes data. And then we have say just a random numbers in a matrix, in a 2x4 matrix. And then this is the way you create a summarized experiment object. You just say the essays is this expression matrix. The row data or the features data is the GRanges object that we just created. It was also 2 rows. And then the column data would be the phenotype data that we just created. And then you would see that you have this nice structure that ties together all these kinds of all the three different types of data. And then anyone can access this object by saying, on this summarized experiment object, give me the assay, and then it gives you the expression matrix that you added. Give me the row data. It gives you the features data that you also added. Or if you want more specifically from the row data, you want the GRanges object with the sequence, with the coordinates that you created. You can also use this function. And then if you want the phenotypes data, you can just say call data. So I see that there are a lot of names of functions here. So you probably don't need to worry about the specific names because you can always Google it, or you can look at the vignettes of the summarized experiment object, the summarized experiment package. But the idea here is that I'm trying to explain how Bioconductor can be used to code an experimental design and include it and like formalize your data that in a way that you can share or you can compute on later on. So another package here is called the R-Track layer. An R-Track layer is basically designed to help you import and export different types of files, which is, I know it's a lot. It can be a very tedious problem in bioinformatics that there are different types and different file types and different formats, and it can be very difficult to understand without looking at the documentation. And very often the documentation of these files also are very problematic. So here the Bioconductor allows you to just read in the file if you know the extension of the file, and if you know that there is a function that exists in R-Track layer that can read in the file, and then you can deal with it as you would with an R object, if you are already familiar with R. So the one that we will be using today is the bit file, which is what we use to represent the ChIP-seq data or the ChIP peaks. And it's very analogous to GRanges object. It's 4 or more columns. The first one would be the sequence name or the chromosome name, the start side, the end side, and then the strand, and then you can add any arbitrary number of columns as metadata. And then once you call import .bid on a regions file or on the PID file, the PID file would be read automatically in a granges object, which is very convenient in this case. One of the annotation packages that we'll be using is called it's an organism package. In this case, the Homo sapiens package. And this is also very convenient because this package that contains a lot of different identifiers. So it's also another problem in bioinformatics that different people use different identifiers. So some would use the gene sample, some would use the ensemble, some would use the entree gene IDs. So we really want to be able to map between all of those. And Bioconductor provide a very convenient way to do that. So there is a function called columns, which you call on the organism package or the object in this case, and then it gives you all the identifiers that you can map to and from. And in the next section, I will show how to do that. Another annotation package which is very useful is the taxonomy, one of the taxonomy packages in Bioconductor, and they have for each organism and each version of the genome, they have a specific package. So this one for the HG19 human genome, and you can also call columns on this object to see what kind of information it contains. In this case, it's all about genomic locations. So you can see the coding regions, chromosome name, you can see the exons, the transcripts. You can see the IDs of the gene IDs, which chromosome they come from, what is the exon name, or what's the transcript name. So it's very useful to be able to work with this. So to interact with those last two packages, you need to use this annotation DBI. And it has a very simple interface. It's a single function called select. You give it the name of the organism that you just, the name of the object. In this case, each package has an object with the same name. So you just load the library and then assign it to a simpler name, or you can just use the same name of the object itself. And then you pass it to select, and then pass it the keys that you want to, like say, for example, in this case, I want to say I have the gene symbols and I want the entree IDs. So I pass it the keys as the gene symbols that I have. And I tell it that the key type that I just passed is simples, and I want the entree IDs, and this is what it does automatically. So this also can be very powerful. Another type of information that we can extract from these packages is the genomic coordinates. So in this case, I'm using the second package, the taxonomy package, this one. I also assign the object that is contained in the package to the TXDB R object. And then I'm passing it to a function called genes from the genomics feature, the genomic features package. And then I'm saying I want the genes in this organism. More specifically, I can filter by the entree IDs of the genes that I'm interested in. And then it gives me the start side, the end side, the strand of these regions or of these genes that I'm interested in. Another convenient package here is to ask for promoters. So I gave it the genes that I just took, and then I say they want 3,000 bases upstream, and then it will do that automatically and give you back the same object, but the ranges are resized to include only 3,000 base pairs upstream from the transcription start site. And then we will talk about the target package for the rest of the presentation. I think I can stop here a little bit and see if people have questions.
I think there is no question for now.
Okay.
Please, dear participants, don't hesitate to leave questions in the chat.
There is one question. So Abdullah is asking if there are any Arabic websites explaining the R and bioinformatics.
Unfortunately, I can't comment on the Arabic content on bioinformatics on the internet, but to be honest, I haven't, I did not come across a lot of content The other thing also is it would be very difficult to like move to like write, but because most of the time, like say for example, if I'm writing a function name or a function or a package name, it would be really hard to write Arabic and English at the same time. So yeah, I don't know like the answer to the question. Maybe there is. If there is, I would be glad to see and to know that some people are writing contents in Arabic.
Thank you, Mahmoud.
So we'll let you know, Dr. Mahmoud, if any question will come along. Thank you.
Okay, yeah, so I will move on to the next section now. Okay, so in this section, I want to introduce the target package and this and the method that I'm using to integrate those two types of data. So I will start by motivating the problem just very briefly. So we have, if like we know that the binding of transcription factors to the gene promoters can either induce or repress the gene expression. And we also know that usually transcription factors don't work alone. So they bind to the same site with other factors and cofactors and other DNA binding proteins, and usually they function in complexes. So the integration of these of different types of data, or even of data from different transcription factors or transcription factors and proteins, become very important if we want to understand the nature of these interactions. So what I'm trying to do here is to use the overlap between the binding peaks of two transcription factors to find the shared or the common targets of two or more transcription factors, and then use the gene expression to model the effect of this binding on the shared genes. And more formally, the question would be limited to, are those two factors, if they have a common target, do they cooperate together or compete? And then the definition of cooperation and competition here is functional. So for example, if two factors produce the same effect on the shared target, then they are cooperating. If they produce opposite effects, then they are competing. So the most straightforward way to do that is to run an experiment where you perturb one factor separately, and then you perturb them together and see the difference between the two data sets on the two data points on the gene expression. However, most of the data that exists is just a single perturbation, and simultaneous perturbations also have other problems that are created from the nature of the indirect interactions between these factors. So the first thing would be to model the potential of a factor to be a transcription factor, or to be to affect the transcription of a gene. And using a model suggested by Wang et al. In 2013, the paper suggested this model where we model the binding sites as, or we model the binding potential as the distances from the transcription start site relative to the distance that we will include, relative to the region that we would include. And then we discount these distances in using this decay function, so that the shape of the score, the shape of the function of the scores would eventually approximate what you would see empirically. So to visualize this idea, I wrote this very simple code here. So imagine we have 100 peaks, and then using this function that I just showed here, code it in R, and then to discount the distances, and then plot the distance on the x-axis, and then plot the score that we would eventually get on the y-axis. So you get this shape. So it's non-linear, but the key point here is that you have the peaks that are very close to the transcription start site, meaning the distance is small, would have a very high score. And then as you move away from the transcription start site, the scores will be discounted, or you will get lower scores. So to deal with the issue with a single feature, a gene or a promoter having multiple peaks, then we also sum those together to move from a peak score to a gene score or a feature score. So the number that you will get for a gene would be the sum of the scores of all the peaks that occur within this region. So to visualize this also, imagine again that you have 100 peaks, but those 100 peaks don't belong to 100 different genes. Some promoters or some regions have more than one peak. Then for those, you take the sum of the peak scores. So most likely you will end up with something like this, like the peaks, the regions that have more than one peak would usually have higher score, which is also what was suggested by some empirical research, that the transcription start sites that has multiple binding sites on the region, as identified more specifically by ChIP-seq, would usually be true targets or be stronger targets for this particular transcription factor. And then, so right now we have two pieces of information. One of them is the regulatory potential based on how far or close the transcription factor bind to a particular region. And then the other piece of information would be the change in gene expression as a result of perturbing this particular transcription factor, either by knockdown or by knockdown or by overexpression. So what we do here is take those two pieces of information and then rank the genes based on them, and then multiply these ranks, this gives us something called the rank product, which is the gene score that we just calculated multiplied by gene statistics. So an example of these gene statistics would be the fold change or the t-statistics from differential expression. We use the ranks to make the data more robust, to make the calculation more robust. And then the final rank or the rank product would be the rank of how important this gene is, or how likely this gene to be a true target. And then it will also give you an idea about the direction of the effect of the binding of this particular transcription factor on gene. Okay, so the idea here is that, again, I'm trying to visualize to show this visually. If you have a binding, if you calculated scores for the binding for a specific transcription factor on multiple regions, you put it in the x-axis, and then you put the t-statistics or the fold change on the y-axis. So we'll end up with a rank product and do the calculation as I just explained by multiplying the ranks and use the rank product. So here I'm showing that the minimum, the region or the region that has the lowest rank product would have a lower score based on the distance, and then would have very low change in expression as a result of the perturbation. And then the genes that have very high score and very high fold change would be ranked very high. So I extended this idea a little bit to see if the interaction between two factors, to see if we can model the interaction between two factors from independent data sets. So the independent data, so for example, here, if we have a factor X and a factor Y, then we perturb each separately, say using a knockdown or overexpression, and then calculate the differential expression and get the t-statistics or the fold change. By multiplying the two of them, we get a new term, I'm calling it the regulatory interaction. Then we use this regulatory interaction we use this regulatory interaction as a substitute for the gene statistics that we used in the previous step, and then multiply it by the rank of the regulatory potential, and then get a very similar ranking to the one that we explained. But the difference here is, because we're using the regulatory interaction for two genes, we have one more piece of information. We know that because the product of the multiplying the gene statistics coming from two different numbers, first of all, the magnitude of the number should increase if they're both increasing in the same direction. The other thing is the sign of the interaction. So if I have a negative fold change and a positive fold change, then likely the factors produce different effect or opposite effect. But if I have two positives or two negatives, then the factors produce cooperate or produce cooperative. Their effect is cooperative on this shared target. So it will be visually like having 4 different quadrants, and then on the opposite sides you will get opposite action. So the ones with negative and positive change, positive fold change, or T statistics, will be competing factors, and the ones with the same sign, either negative or positive, will be cooperative. So the next step would be to try to aggregate the effect. So if we stop here, the problem is most factors, if you looked at the overlap of 2 factors, eventually we'll get some overlap. And also if you knock them down separately and you try to do this calculation, you will see that they will produce many cooperative effects and many competitive effects. So we want to be able to quantitatively figure out on aggregate or what is the most dominant function that the interaction of those 2 factors produce. So I'm using something called the empirical cumulative distribution function, or the ECDF, which tells you for if we divide all the genes, all the genes or all the regions where the 2 factors interact into the ones, say, with the positive sign and the ones with the 2 positive signs and ones with 1 positive and 1 negative. So the ones where the genes compete or the genes interact. Then we have 2 categories of genes, and we calculate the empirical distribution functions of those 2 categories. And then it tells you the proportion of genes in this particular category that is ranked at or higher rank than a particular value. And then what you get is a curve like that. And then you can compare this for each category. And then you can compare this curve to a theoretical distribution by asking, if these interactions are random, how should the curve look like? And then if your curve look differently, then it tells you where do the interactions tend to be, on the positive side or the negative side. The other option would be to compare 2 curves, as we will do later on in the presentation. Okay. I want to show a diagram of this workflow or of this method, just to summarize what kind of data do we need and what do we do with it and what do we get at the end. So we need 2 different sets of data. One of them is the binding data, for example, from ChIP-seq experiment, for 2 objects separately. Then we get the binding peaks, and then we find the overlap between them. The overlap would be the potential shared targets or common targets. And then we have gene expression data, where we perturbed each factor separately, and then we get any signed statistics. It could be the fold change or the T statistics. We multiply them together to get the statistics product, and then use the rank from the regulatory potential. We calculate it using this the decay function, and then use the rank from the statistics products of the differential expression statistics. And then the multiplication of the 2 gives you a final rank of how important this particular gene is in terms of the interaction between the 2 factors. So the results of the multiplication would be the magnitude, how strong the interaction is, and the sign, how, like, the direction of the interaction. Okay, so this analysis is implemented in 4 different functions in the target package. So the first one is called associated peaks, and this one calculates the peak score and assign it to the nearest gene. And the next one is called direct targets, which what it does is take the regulatory potential, calculate it here, or the peak scores, aggregate it for genes, and then multiplied by the statistics from the differential expression, and rank the genes in one big rank to see what are the most important genes and what are the least important. And then there are two helper functions to test and plot the predictions that we made in this analysis. So there are a lot of ways to integrate gene expression and DNA binding data, and there are different approaches with different goals in mind. So these are just 3 examples of other R packages that does this or is something similar. So there is a package called TRM, and what it does is define transcription regulatory modules as complexes of transcription factors and cofactors, and also protein-protein interactions data. So what they do here is not ask empirically which factors have the same effect or not, just use known protein-protein interactions and make out of this information, they can make what's called the TRM, or the transcription regulatory module. So you can also check this out to see if it could be helpful for you, for those of you who are interested in integrating data. There is also this other package called TFEA, and what it does is pull down a lot of ChIP-seq data and then analyze it and then process it and then make this available as a database, and then people can just query it. That could be very useful for people who are struggling with processing and pre-processing ChIP-seq data, because that can be resource intensive and it can be a bit tedious if people are just starting. So we can just check out this kind of work to get familiar with the data itself, the processed data, before you worry about doing alignment and counting and peak calling and so on. The transcriptor package does something also very interesting, which in this package they use ChIP-seq data to identify transcripts. So in this case, they're not particularly using transcription factors, but they use DNA binding proteins, or it's possible also to do it using histone modification ChIP-seq data to identify the transcripts themselves, and then use the RNA-seq data and then count the RNA-seq reads within these regions or within these transcripts that you just identified, which is very interesting way of using the 2 types of data. So there are limitations to this kind of analysis. The one I'm presenting in Target is that we need a very specific set of data. So we need 2 sets of data for 2 transcription factors. Usually it has to be in the same cell line, because these kind of interactions are really very cell specific or tissue specific. So I'm not sure how reliable would be the output if you use data from different cell lines or different tissue. The other thing here is that I'm assuming that the interaction between the two DNA binding factors or proteins is linear, but it might not be always the case. There are specific forms of binding to the DNA that can't be captured using this analysis, which is a limitation of the ChIP-seq, not of the analysis, to be exact. But for example, the first step to find the interaction between 2 factors in my analysis is to find the common peaks or the overlapping peaks. But if for whatever reason those 2 factors interact without binding to the same site, this will not be captured in this kind of analysis. So finally, I want to tell you how to get starting with this analysis. The best way would be to check out the Target package, which is an open source Bioconductor package. And you can use this link to check it out. There is also an interactive Shiny application for people who like this kind of stuff. And it's also available on the web. And you can, if you download the Target package itself, you can run it locally as well. And then finally, the code for this presentation is also available here. Yeah, so the next I will be talking about the use case or the example of those two transcription factors to see how the output of this analysis look like. But maybe I can stop here for a couple of minutes and see if there are any questions.
So we have one question. Do we need control unperturbed data sets to determine the G genes? Then we will know which genes are regulated by the interested regulatory factor. So differentially expressed genes.
Yes, you need the controlled unperturbed data. Because one of the steps is that you need to calculate the differential expression as explained in the question, because you need the t-statistics or the fold change. However, this is not an issue for transcription factors because there are a lot of data sets. Okay, I just posted on the chat a name of a database called knockdf, where they have hundreds of transcription factors in tens of cell lines. So they curated this large data set from Gene Expression Omnibus and Array Express and the Short Read Archive. So if you're struggling with this particular point that maybe you don't have the specific data set, maybe knockdf would be the first place to go, either to find the data set, the publicly available data set, or to just find the process data directly and see which genes go up and which ones go down.
So any other question, please? There is another question. Can we take data sets from any platform recent like Illumina or Nanopore?
Yeah, Illumina for sure. I'm not familiar with Nanopore, but I would imagine there is a way to do that. If you mean for the differential expression, yeah, for sure, because all platforms will give you at the end a value that says this gene is going up or down. But for the binding data, I'm not sure if they use Nanopore for this purpose. Okay, there is, I think, a second part to the question. Do you mean like if you have data from these platforms, can you use the same bioconductor packages? Is this okay? Yes, as long as you could. So what you need is a data frame and a GRanges object, right? As long as you can get your data in these formats, it doesn't matter where they come from. Obviously, it does matter for the biological interpretation, right? But for the package to work, you need the GRanges object for the binding peaks, and you need the data frame for the differential expression. So yes.
So I think about Illumina and Nanopore, does it work for long and short reads or only, it's my question actually, or only short reads?
Well, the chip reads are usually shorter than what you get from Nanopore. So that's why I'm saying, do they do ChIP-seq with Nanopore? I don't know. If someone knows, you can tell us in the chat, and then we can ask to try to talk it through, I guess.
Okay, then no more questions. So thank you.
Okay. Okay, I'll move on to the last part of the presentation. Okay, so this is just a very simple, like typical analysis of a typical analysis to try to find out if two related factors can do interact together or they just work independently. So there are a lot of packages required. So I hope that people who are trying to follow the code or like trying to execute the code are not having problems with installing all these packages. But this is the nature of this kind of analysis, that you have to have to use many packages and to put together different data sets from different sources. Yeah, so it's key to be able to work with different packages in the same document. So the motivation of this problem is that we have two transcription factors called YY1 and YY2, or yin yang 1 and 2. And those two factors are from the same family. So there are both of them are two zinc finger proteins. And then one, YY2, is a retrotranspose of or arose by retrotransposition from the YY1. So they are evolutionarily related. And there are data that suggests that they are also functionally related, as you might imagine. Two transcription factors that came from the same gene would have either work on the same targets, or they would produce similar effects. So using this target analysis, we're asking, do those transcription factors share the same genes, or share all genes or some of the targets that they work on? And what is the consequence of the binding of these factors to the targets, either separately or together? And then like if they do interact, if they do have shared targets, like if the answer to the first question is yes, and the answer to the first question, if the answer to the first question is yes, then what could be the function of this combined binding? So to do this, I'm using a data set I'm using 3 data sets. One of them is microarrays data set of YY1 and 2 knockdown separately in HeLa cells. And then there is a ChIP-seq data set of YY1 in HeLa cells, and another one for YY2. So that gives us the 4 different pieces that we need. We need perturbation for each factor separately, and we need the binding peaks for the 2 factors separately. Well, unfortunately, I'm not going to discuss how to pre-process or process this data, because this is probably the different topic. But we will be working with the process data directly. So the differential expression was calculated by the knockTF, but it's just standard differential expression that you do in Bioconductor using Lima or HR. So if you look on this database, it's very useful for people who are interested in this topic. Then to get the binding peaks, you can check the ChIP atlas, which also have a large curated data set of binding peaks from ChIP-seq data and ChIP also for many transcription factors and DNA binding proteins. So you can get the process data from this data set, but these are the original sources and the papers that came out first. And then I'm using the HG19 human genome to extract genomic coordinates and genomic information. So the 3 parts of this analysis is that we will prepare the sources, the data sources for the target analysis, then predict the specific targets of each individual factor, and then predict the combined function of the 2 factors. Those are the 3 different parts. And I guess the most important for people here today is to try to understand, or to get an intuition about how to move from different data types, or like different file formats, into just the data frame that you need, or just the G-ranges object that you need. So in this case, we'll start by preparing the binding data, which comes in the bed format, and this is downloaded directly from the ChIP atlas. So I haven't modified the file. You can just download this from the ChIP atlas for the transcription factor that you're interested in, and then you use the import bed to read the files, to read each of the 2 files. So we have 2 files for each. We have 1 file for each factor. So in total we have 2. So import bed from the RTrack layer, and then you can say, give me the G-ranges object from the file that you just read, and then assign this to an object called peaks. So now we have the first piece that we need for this analysis. So to look at these peaks, this peak now is a list of G-ranges objects, and then you can use this convenient function called linksys to see how many different peaks in each transcription factor. So we see that there is, like, the YY1, the original transcription factor, have way more binding peaks than YY2. And then this is how the object looks like. So just the standard G-ranges object, the sequence name, the chromosome name, the start site, the end site, And then the strand, in this case, the strand is not recorded, which is also, it's not uncommon for ChIP-seq data to come in this format. And then there are some metadata about the peaks. You can use this convenient function called table to see how many peaks in individual chromosomes, if this is interesting to you. And then obviously you can use this to visualize the coverage of the ChIP-seq data on different chromosomes. And then I'm interested in the width of these peaks. So I'm using the summary function to see what is the distribution of the distances, because we will use this to calculate the peak scores. So here the average distance is about 200 base pairs, which is also what you expect. So to prepare the expression data, if you started the vignette, you would see that the data exists and that you can download the data into that you can download the TSV files or the text files for the expression data or the differential expression. It's exactly the output that you would get from Lima. In this case, I'm only interested in those 4 columns. So that would be the 3 columns, sorry. And then that would be the gene name, the fold change, and the p-value, right? And then I have the 2 for, I'm filtering by YY1 and YY2, the ones that I'm interested in. And then read this in a data frame or a table, assign this to express or expression. And this would be the second piece that we need. So in this case, I'm checking the number of rows for each data frame or each table. And in this case, they have the exact same number because they record the same features. And just to double check, I'm checking whether the gene names in 1 is the same as gene names in 2. And then just to get an idea about the significance and the direction of the interaction of the fold change for the factors separately, we're seeing what is positive fold change and what is negative, and what is below a certain cutoff and what is above. And then we do that for the 2 factors. So if you look at the numbers, you would see that the knockdown of either factor changes a lot of genes, changes the expression of a lot of genes. And these changes are either direction, which is not uncommon in this kind of data, in these kind of data sets. The changes that result from the knockdown we will see that they are correlated, as I will show in the next figure. So this is exactly the case that I'm looking at. The 2 factors have wide change. They produce a lot of changes in the gene expression of their targets. And then these changes are somehow correlated. So we can actually see whether this correlation, or like we can even ask how strong the correlation is. Do they go to the same direction or the opposite directions? So here I'm making a simple graph for each factor to show the fold change and then show the significance level, taking the log 10 to just make the data easier to say. This is called volcano plot. So in this case, I'm making volcano plots for YY1 and YY2. So you see there are a lot of changes on either direction. And then here I put the fold change for one factor and here fold change for the other factor. And you can see that they are strongly correlated. In this case, you can calculate the correlation using the curve function in base R. And it is a very strong correlation. So next would be to prepare the genome annotation. And this is where Bioconductor really shines, because you got pretty much everything you need, and there are very convenient interfaces. So what I will be needing here is the entree genes, gene IDs, and then I have the simples, right? So I just give it the object from the organism package called org-hsegdb, and then give it the keys as the gene names. In this case, I'm using the gene names for the first factor, but they are the same. And then say, I want the entree IDs. And by the way, the key type that I just passed is the simples. And then what you get is a data frame with one column is the samples and one column is on tray IDs. And then I'm removing the missing values because not every gene sample can map uniquely to an on-tray ID. And then I decided here to use only 100,000 base pair or 100K base pair upstream from the transcripts, because the binding of any any binding peaks that is very far from the transcription start site will be likely irrelevant here, because even if this is functional, it's unlikely to be function as a transcription factor. But I understand that there are a lot of exceptions to this. We're just using we're just making an analysis decision in this case. So I'm using this convenient function called promoters to take the taxonomy object for Homo sapien and then extract 100,000 base pair upstream of the transcription start site. And then I'm matching the gene IDs from here, from this object to the on-tray IDs because the order is different. And then I'm subsetting, removing the, I'm adding the gene samples to the object, the genome object that I just created. So this was the third piece of input that we need. So the first piece was the peaks, the binding peaks for the two factors, then the differential expression for the two factors, then the genome information or annotation for the features or the targets. So this is the object that we created using the select interface. The samples in one column and the on-tray IDs in one column. And then the taxonomy object we used to extract the genomic annotation. And then here for each of the features that we recorded, so remember we only had the gene sample, but from that we could get the gene ID or the on-tray ID, and then from that we could get the transcripts, and then we can, for each transcript, get chromosome name, start site, and the end site, and also the strand. So we need this information because eventually we want to calculate the distance of the binding peaks from the transcription start site. Okay, so now we need to merge, because remember we have differential expression. So differential expression is recorded by the gene sample. So between the genome information and the differential expression data frames, we have a shared column. So we can use this to merge the two objects. What I'm doing here, match the gene names from the gene samples from the genome object, and then match the gene samples from the differential expression objects, and then add these to the to a new genomic range as metadata. And this is how the object look like. So the first part is exactly the same like genome. And it's the chromosome name, the start, the end, the strand, and then some other information about the transcript IDs and name. And then this was the shared column. And then from because we merged the two, then this is the differential expression information. So that was the gene name, the fold change, and then the p-value. So right now we can calculate the width, or like we can check out if this happened, if what we did is correct by saying what is the average width, or what's the width of the regions that we just extracted, because remember we only wanted 100K around the transcription start site. So we will do the analysis first for individual factors. So in this case, we have the peaks, the peaks and the regions. So what we want to do first is calculate is to call the associated peaks. So this function has only 3 arguments. The first argument would be the peaks object, and then the second argument would be the regions object, and then the third would be just to tell us to tell the function to tell the function which column to use as names for these peaks. Oh, sorry, which column to use to aggregate the peaks. Because remember, like different some transcripts or like some genes would have more than one peak. So at some point we need to aggregate the peak scores. So we tell it which of the columns in this object is the one to use to aggregate the function. And then what associated peak does is return the same object with it and add to it some new columns. So in this case, it added the regions where this particular peak was assigned, and then it added the distance of this peak to the transcription start site, and then the peak score that is based on the distance, right? And remember, we're doing this for the two factors. So that's why we're using this map2 function, right? Because we're doing the calculation for the factors individually or separately. So the second object also is identical to it in format, at least. And then you get also the assignment of the peaks to the factors, the peaks to the genes or the features, and then the distances from the transcription site, and then the peak score. So we see here that those are the original column names, and this is what associated peaks returns. And then it's only 3 extra columns: the assigned region, the distance, and the peak score. Okay, so direct targets have very similar interface. Also the peaks object, the regions object, and then the column to use to aggregate effect the peak scores. And then the stats column is which differential expression statistics to use, and it also has to be a column in the regions object. And then the output is also goes the same way. So it's the original object that you gave here, the peaks, merged by the regions, and then some extra columns. So what we have here is the peaks, and then the scores that we calculated from before, and then the rank of the score, and then the differential expression statistics, and then the rank of it, and then the rank product of those two ranks. So the score rank and then the stats rank. And this would be the final rank that we would use to see how important the genes, individual genes are. And again, we're doing this for the two factors separately. So how does the data look like at the end? Okay, so it would be really hard to look at these values one by one and compare it, right? So I'm using this empirical distribution function to aggregate the effect of the factors, because what we're interested in at the end is to say, does this factor induce or repress the gene expression? So to do that, I'm dividing all the genes based on the fold change into 3 groups. Right? And then I'm just color coding these groups and naming them. So the very lowest group would be the genes that have negative fold change, and those would be downregulated as a result of the knockdown of the gene. And then the ones in the middle that has very small fold change, either positive or negative, we'll just say it's non-regulated. And then the ones that has positive fold change will be upregulated. And then we pass the score that we got from direct targets. Remember, that was just one column here, the score rank, otherwise also known as the regulatory potential, and then give it the groups that we just made here from the differential expression statistics. And then what it gives you is those two figures. So in this case, it's saying that there are two distribution functions for the scores, the regulatory potential that we calculated for each factor, right? So you see here that many genes, or the genes that are downregulated, are more numerous, and they also have higher ranks than the upregulated genes. And remember, because this is the result of the knockdown of these factors, so the effect should be flipped. And then you also see that the YY1, YY2 haven't the opposite pattern. So more genes are upregulated than downregulated. So this was for the factors individually. So there is a way to statistically test whether those curves are different or not. Sometimes it's really obvious, but sometimes the curves wouldn't be. It wouldn't be that obvious, especially if the curve is not. The curve has like turns at different points. So there is a statistical test called KS test, which you get to use it. You can. There is also a helper function or wrapper function in the target package that has very similar interface to predict to plot prediction. You pass it the ranks, the final ranks from the output of direct targets, and then you give it the groups that you created, and then tell it what to compare. So in this case, we're comparing the downregulated to the upregulated, and then it gives you a t-statistics of the distance between the two curves, and then calculates a p-value. Okay, so now I want to move on to the final part, which is predicting the interaction of the two factors on the shared targets of the two factors. So there are two main differences from predicting the function of individual factors. I mean, other than the input data that you use. So the first one would be to use something like subset by overlap, which takes two G-ranges objects and then gives you the overlap between them. Because remember, we recorded the data by saying this peak is in chromosome X, starts here and it ends here. So if we have two peaks, if we have this kind of information for two peaks, we can figure out whether they are overlapping or not. So in this case, I'm interested in the shared targets of those two factors. So if they bind to the same site, at least in theory, we should get overlapping binding peaks. So this is what I do here: find the overlap between the peaks of the two factors. And then we need to reformat the differential expression data frames, because we had the list of data frames. In this case, we need them both in the same data frame. So I'm using this function called pivot wider, which takes one or two columns and then spread them into two separate columns by a third column. Okay, and then this is just like a routine to match the genes in the genome object, match the gene samples in the genome object to the object that we just created from the data frames. And then just remove the genes that are not, just to make sure that the genes in both are the same, and then add this new data frame to the genome information as metadata. And then call common peaks the same way, no change. The only difference here is that we're passing to it the common peaks, not the peaks of the individual factors. And then we're giving it the new object that we created that has the differential expression statistics for the two factors, not just for the one factor individually. Direct targets, the only difference here in calling direct targets is you have to, instead of passing it one stats column for the full change for the knockdown of the factor, you pass it two columns or two column names for the knockdown of the first factor and also the knockdown of the second factor, right? Because those will be multiplied together to calculate the regulatory interaction term. So the output of associated peaks would be the same. And then direct target would have also the same structure, but the difference here is stats is not just the fold change, it's the multiplication of the fold change for the two factors. And then the rank also would be, the rank also would be the rank based on the interaction between the two factors and their binding to common targets. So the idea here is that if this term, this stats, there is a multiplication of the fold change from the two factors, first of all, the magnitude of this term would tell you how strong or how strong interaction between the two factors, and the sign will tell you the direction. So if two factors have opposite fold change, one positive, one negative, you will get competing factors. And then if the two factors have the same sign, then there will be more of the cooperative nature, at least functionally. And then I'm using the cumulative distribution function to divide them to make the same curve to ask what is the aggregate function. In this case, the only difference here is that when I divided the genes into groups, I just called them competitive and cooperative, based on the idea that the factors that are that have the same sign is competitive, is cooperative, and the factors that have different signs is competitive. In this case, because you have just two signs, any positive interaction would be cooperative interaction. Obviously, because if you multiply like minus by minus, then it will be positive. So these are the cooperative interactions. And then the competitive ones are the ones that have negative signs, one negative and one positive signs. So they will be at the bottom of the list. So using these groups, again, you pass it to the plot predictions with the ranks, and then it gives you this function, those two curves or three curves, one for the non-interacting, one for the genes where there is no significant change, and one for the genes where there is competitive change. In this case, the green one, and then one for the genes where there is cooperation. And then by looking at the distance between the two of them, you can see that the cooperative, the genes where the two factors cooperate together is rank higher than the ones where they compete together. However, if the number of factors, the number of genes where there is a strong competition is much bigger. And then also at the very beginning, like the strongest targets are actually competitive targets for the two factors. So this is a kind of observation that you can make from this data, or this is the kind of conclusion that you can get from this data. And then this hopefully would give you an idea what to do next if you want to dig deeper into what is the difference between the factors where the two compete or the two cooperate. You can take like for example, you can take the top 100 peaks from competitive factors and the top 100 peaks from cooperative factors and do something like motif analysis to see if specific motifs are associated with the competition or the cooperation. Okay. And then at the end, I'm using this KS test again to test the difference between the two curves and see whether there are significant difference. There is a significant difference between the two curves or not. So to summarize this here, I'm presenting a workflow to predict the direct interaction between two factors by integrating their gene expression and DNA binding data. The target package is based on an algorithm called Beta, which was implemented in Python. And Beta was designed to work for individual factors. So I just made a little bit, I extended it a little bit to work for those two transcription factors. So the conclusion here, if you want it in one single line, is that if you have gene expression data and DNA binding data for two factors, then use the DNA binding data to get the common peaks or the overlapping peaks. So those would be the shared targets, and then multiply the differential expression statistics by each other, right? And then you can model this effect using the decay function that I just showed, and then rank your genes, then dig deeper into what's happening on the top of the list or at the bottom of the list. Okay, thank you so much for listening. I've been talking for a while now, so I'm looking forward to hear back from you.
Let's see if there are questions.
Thank you so much, Dr. Mahmoud. That was so awesome to learn from the valuable our knowledge that you shared with us. Thank you so much.
Yeah, it was really amazing. Very clear.
Yeah. the way how he explains. Yes.
So please, are there any questions?
Thank you so much, Betul, for joining us. So just to mention, Betul is from Our Ladies, Saudi Arabia. She's the founder.
Okay. Yeah,
So,
good to see you here.
so we have here a question from Laura. She said, thank you, that was really helpful and interesting. I was wondering if there are any particular resources or packages you recommend for single cell analysis of this type.
Oh, okay. Honestly, I'm not so familiar with single cell, but what you can do is look for something called BioC views, and then views are like keywords that you can use in Bioconductor. So if you type single cell, it will show you all the packages that uses that is concerned with or like deals with the single cell data. So I guess use BioC views to see which packages you can use for a single cell.
Thank you very much. Okay, so maybe we can show about how to download the teaching material with Docker. So we can, the first part, so then
okay.
everyone
can go back to
use Docker.
Yeah, well, actually, I'm really sorry because I'm using just my laptop, so I can't run Docker here. But I will show you the code and the links. So if people want to execute the code on their machines, they can do that.
Yeah. So how I'm actually curious about if you have like teaching materials, how to make a bioconductor or a package, an R package from them. So,
oh, okay.
Yeah. Is there any package or function do you use for that? Because
okay,
then
so
you can, sorry.
Oh, no, that's all right. Yeah, go on.
So once you have your package, then you use you make a Docker image from that package, or how it works?
Yeah, so the reason I'm using Docker in this case is that it's there are a lot of packages that are used in this analysis. So what Docker does is use it, take an operating system image like Ubuntu in this case, and then install R and install the packages, and then make this image fix it, and it's locked for the version numbers, right? Like the particular version of this software. If other people want to use it, instead of downloading the packages and installing them one by one, they can pull the image itself, right? And then once you have this image, you can make an instance of it, which is just like an RStudio server backend to work on and execute your code. So I find it very useful, not just for workflows, but even for data analysis, to just lock in the environment. Because otherwise, if any change happens in a package upstream, it will break your code and then make it unreproducible.
Thank you very much. Really interesting. So if you can show how to run the Docker image, it will be really helpful. Thank you.
Okay, I think there is another question in the chat.
Yes. So the question is it possible to compare three factors through the regulatory, maybe much more complicated and hard to interpret?
Exactly, yeah. The thing is, because I'm relying on the sign to see if the two factors, the direction of the interaction, so multiplying three of them would not make sense. The other thing is like zero values or very small values. would also skew the whole thing, right? But you could use like something more involved, like a linear model or something, and then make an interaction term and then model the data using the same way. But I haven't really thought too deeply about this, because to be honest, also I was developing this particular tool for like a particular application that I was interested in. But yeah, it would be interesting to see if I'm sure there is a way, specifically using something like a linear model or like interaction terms to do that. But I haven't done that, to be honest. So the target package does not handle more than 2 factors.
Thank you very much.
Okay. I'm going to share the screen again and just go through how to download the Docker image. Okay, so the 2 commands that you need for to do that are the ones here. So I mean, assume that you already installed Docker, which is a thing on its own, especially if you're using Windows. But yeah, so you say Docker pull, which is very similar to like Git or GitHub pull, right? You just like pull down a local image of this particular repository, which is an image that I created before. And then you say pull run or Docker run to this image, and then you just give it a port like to the browser, and then give it a password for RStudio server, just like you would use RStudio server. And then yeah, once you do that, you can use your browser, go to this URL, I guess you call it a URL, yeah, in the browser, and then you will have a local instance of it, and then so the Docker itself is running in the background through the browser, and then you can run it just like you would run RStudio. Is that clear, or people need a little more on this part?
Please don't hesitate to tell us that in the chat.
Yeah, so this is how the Docker image itself look like. So well, I guess this is not how it looks like. Okay, so yeah, the Docker image is very simple. You just add an operating system, Ubuntu, and then R and RStudio, and then install the packages just like you would install on your local machine. And then just keep it if you want to use it locally, or push it to Docker Hub if you want to share it with other people.
I hope that that's clear for you. So if you are done with the questions, thank you all for joining us today, and special thanks to Dr. Mahmoud again for animating this incredible workshop. We'll be glad to share with you all the workshop materials later, including the recording video. And stay tuned to our next meetups. Goodbye.